back to some basics. This is like how we always used to think about soil, and this is how it's taught at university, still taught at university like this, that, you know, the soil is the foundation of everything and plants grow in soil, that's obvious, and animals consume plants. And the problem with having that kind of a structure in our mind, I mean, that's how we see it out there, is there's soil and then there's plants and then there's animals. But if our plants aren't performing as they'd like, or our animals aren't performing as we like, we think there's something wrong with the soil that we need to fix by taking a soil sample, sending it off to the lab, getting it analysed, trying to find out what's in it, and trying to correct the so-called deficiencies. The problem with that is that the soil that you send to the lab is going to get exposed to chemicals it's never ever exposed to in the real world, and the extraction process bears no resemblance to what actually happens in soil in terms of how microbes obtain nutrients. And the assumption still is that plant roots are just going to suck nutrients up out of the soil passively, and that there's not going to be any microbial intermediaries there, that there's not going to be transformations by microbes. So what you see on a soil test is almost totally irrelevant, apart from perhaps your carbon level, which is important. So that's the problem with actually looking at it like that. We now know, I've got one of these. We now know that humus-rich topsoil, which is what we want, is a product. It doesn't just get there by itself. It's a product of photosynthesis, so we obviously have to have green plants, and a diversity of those, and microbial resynthesis. So the microbes are so important in that process. So photosynthesis is the basis of our triangle. Soil is really just at the top. It's a product. We've got the whole thing upside down when we look at it like this, because couldn't be further from the truth. We've got it, 180 degrees out. We need plants, lots of different kinds of plants. We need to be looking at their roots. What do the roots look like? Are there lots of microbes around the roots? And when that's happening, when we see things actually happening around plant roots, then we know we're forming soil, which is what that plant is. And then this morning I talked about, that we need to understand what's actually going on inside that riser sheath. Like, what are the conversations that are happening in there, and why is that life around that young seedling so important, and the life around a seed that's just germinating. So the conversation that we had about what happens if you put insecticide and fungicide and high analysis fertilisers near a seed, what conversations are we interrupting? Why is that important that we don't, why is it important that we don't do that? So something I mentioned very briefly this morning, but I didn't go into it in any detail, was quorum sensing. So in human society, a quorum is just the minimum number of people that you have to have in an organisation for some kind of business to be transacted. So if we had the Mackay Cricket Club or something, and there were, say, 10 members on the committee, and the quorum for that committee might be 6. So you have to have 6 members of that committee have to turn up for a meeting in order for some kind of business to be transacted. And they say, well, we're going to put this proposal forward. We're going to spend $10,000 upgrading the cricket pitch, and we're going to have a meeting about it. They have a meeting and only 3 people come. So even though they're all committee members and all perfectly capable really of making a decision about whether they spend that $10,000 or not, if there is not a quorum for that group, they cannot make that decision, and that business cannot be transacted. So even though 3 committee members came to the meeting, it was quite clear that the cricket pitch needed to be upgraded, and they'd all be perfectly capable of, agreeing to spend that $10,000, they haven't reached a quorum. So any of you who've ever belonged to any kind of an organisation, you know what a quorum is. So if they recall that meeting and 7 people come, they've reached their quorum, they can sign the cheque, they can say, yes, we can spend that $10,000. Well, it works exactly the same in microbial communities. This morning I said, one single bacterium can't do anything, but a certain number of them working together can, and they coordinate their behaviour and they communicate with each other. And when they reach a quorum, then they switch on their genes for doing that job, whatever it might be, fixing nitrogen or something. There's millions, literally, or probably thousands, maybe I'll be a bit more. There's definitely thousands of different kinds of bacteria in the soil that can fix nitrogen. Do not limit yourself to thinking it's just rhizobia in association with legumes. 99.9% of them cannot be cultured in the laboratory, and so we can't multiply them and sell them, so there's no commercial benefit to knowing what they are, so they're largely ignored. But they're what we call non-culturable, but they're there in the soil. They have the nitrogen-fixing enzymes, nitrogenase, and they're quite capable of moving from the soil into your plants through biological induction. But they will only do that when they reach a quorum. So we have to be very conscious of not only what kinds of microbes are there, but how many of them are there. Because all microbial groups, all bacteria, all fungi, all archaea, and probably most protists actually work on quorum sensing. That is the means by which they communicate. And they need to know how many of them are there, and when they are sufficient of them, they can switch on their genes and they can change genetic expression in the plant. They can change genetic expression in the animal if they're in the rumen, and the microbes in our gut can change the expression of our genes. That's how powerful they are. And at the moment, we have got a lot of our genes switched off because our gut microbiomes are not diverse enough. So when they mapped the human genome, they found that what they called 92% of our DNA was junk DNA. We're using 8% of our DNA. So what's the other 92% for? Well, maybe the other 92%'s just been switched off. So have a think about that. Because we know that microbes in our gut can actually affect our DNA, and that's the science. That's not my opinion. So in the microbial world, the term quorum sensing, which is just abbreviated usually just to QS, it's density-dependent coordinated behaviour. Density-dependent means you have to have a certain number of them there. And then it regulates gene expression. It is incredibly powerful for your plants, your animals, and also for people. So it's coordinated, and it occurs in every single species of bacteria, archaea, fungi and virus that we're aware of. So for example, if you just looked at, it's mostly been studied in pathogens, unfortunately, but I guess the thing is that, you know, we're as humans, we're interested in what attacks us and what tries to kill us. So there's been a lot of work, for example, with viruses, like say the flu virus. So if you breathe in a flu virus or a couple of them, they can't possibly dominate you. They can't possibly make you sick. And they're going to have to multiply up in your body to reach sufficient population density to actually have an effect on a big thing like a human being. So while they're in that process of multiplying up, they've got all their genes, all their virulence genes turned off. Your body has no idea it has a flu virus in it because they're not even expressing themselves as flu virus. They're just like benign little viruses just multiplying, and they're talking to each other all the time, communicating using quorum sensing signals called autoinducers. Again, this is a subject we could talk all day about quorum sensing and autoinducers and how these guys work, because it's extremely, it's fascinating really how it works. When they reach a certain population in your body, and they know by talking to each other that there are enough of them to actually overpower you, they will go click, just like when we've got our 7 people or something has come to the meeting. Yes, we can make that decision. We are now going to overpower this human being, and they switch on their virulence genes. And you can read all about that in the literature. There's heaps about it. And then, within a few hours or something, you're going to feel terrible. You're going to have headaches, aches and pains. And, I mean, sometimes if you get the flu really badly, you go down within hours. You can't even walk. If any of you have ever had flu come on like that, you know what it feels like. So that tiny little virus that you can't even see, because there's millions of them, they've overpowered you, but you didn't know you had it until it reached a quorum. So this kind of thing happens in the soil, but in a positive way, when bacteria, viruses, archaea, fungi, when they reach a quorum, they can switch on their genes to do a whole lot of positive things. Like the literature is mostly about the pathogens and about the negative things, but there's lots of positive things, very positive things that happen in soil, like those examples we saw of resilience in a drought. Like there's no water around, and yet those diverse plant communities are like, we're not suffering from any water stress. So how are they dealing with that? Well, they switched on their genes for drought tolerance. Who did that? The microbes did that. Why did the microbes do that? Because those plants are still photosynthesizing, feeding the microbes, right? Microbes aren't stupid. It's really, when you start joining the dots, you can see how these things work, but you have to look at it in a holistic sense. So I love that word holism that Tegan was talking about, holistic. We really have to think about everything holistically. So I'm not going to go into the details about quorum sensing, other than to say that every single species of bacteria or archaea, fungi, whatever it might be, has its own unique signal, so it knows how many of itself there are, because mostly with bacteria it is how many of itself, because they just keep dividing and dividing and dividing and splitting in half and producing more and more of their own things. So they have a unique signal, which is called an autoinducer, but I will mention autoinducers a little bit more later. And so when the concentration of autoinducers in the environment reaches a critical point, that's how they then know, okay, there's enough of us. Like, because as I said before, microbes can't see anything, they can't hear anything, they can't speak to each other. So they communicate using these autoinducers, which are chemical signals that they release into the environment, and every kind of group of microbes have their own sort of groups of autoinducers that they use. And it's a very simple, I suppose, signalling system, in that it's a bit like what happens with the hormones in our bodies. I mentioned like TSH this morning. You have a chemical of some shape is released by a bacteria, has to fit into a receptor that takes exactly that shape. So if it's going to take, what are these ones? I've got 1, 2, 5, something that's got 5 points on it. It's obviously not going to take a square, it's not going to take a triangle, it's not going to take a circle. You know, it's got to be that that shape molecule for it to take it. And then once it's actually fitted into that receptor site, then you'll get genetic expression. So this is what should be happening in the soil all the time. When we look at what happens inside these rhiza sheaths and all the microbes that are living in there and the crosstalk that's going on, inside that environment there would be just like the autoinducers, and the chemical signals that would be in there would be absolutely extraordinary. It'd be like, you know, being on a crowded street at Christmas time or something. That get a bit confusing, you'd think, after a while. But when there's no quorum sensing happening, what we see is what you see there on the left-hand side. They are plant roots where the farmer has used nitrogen fertiliser. So almost every crop of every kind of crop that you'd like to think of in Australia, if we were to go out and collect the roots of those plants and take them back and look at them under the microscope, they'd look like that. In other words, you'd be able to see the roots. That's not good. The one on the right-hand side is where nitrogen fertiliser hasn't been used, and you can't see the roots because they've got all the soil around them. So what you're looking at there on the right-hand side is what happens when the microbes get together and they're using quorum sensing, and they're communicating with each other and collaborating and coordinating their behaviour, because that's incredible coordinated behaviour. You think about it, how do dumb microbes, theoretically dumb microbes, how have they coordinated their behaviour for the bacteria and the fungi and everything to work together and actually glue all those soil particles together and make that rhiza sheath? Because they've created an environment that they can now live inside and do all the things that they need to do. I mean, it's an incredible feat of engineering to change soil structure in the way that they have. And when they don't do that, we see all the sorts of symptoms that we see in agriculture. So that this is wheat, and the one on your right-hand side has had 100 kilos of monoammonium phosphate, MAP, placed under the seed. The seed is right there in the centre of that root system, and it's got virtually no roots coming out from the seed. It's got a very, very short root system, clean roots, and look at the soil underneath it. I mean, you're going to get a plough pan formed under that plant whether you ever used a plough or not. People say, we've got compaction but we don't cultivate. We've got a plough pan and we don't cultivate. And I said, well, you've got a dead pan, that's what you've got, because you haven't got roots going down into the soil and activating the soil. The only thing that can actually transform soil and keep soil alive is plant roots. And if you put high analysis fertiliser under a seed, you're not going to get good plant roots. So the two on your left are grown in a biological system. You can't see the seed because there's so many roots around it. There's so much soil sticking to the roots. They've got much deeper roots, and they're building topsoil. So the wheat is the same age, and it's only like a couple of metres apart on a research station, just the different ways that they're grown. So this compromised soil structure that we get due to excessive nitrogen use is really evident in dry years. I'm just going to show you an example of that, and it's very evident in wet years as well with our waterlogging. So this next photograph, actually, I'm sure some of you will have seen it before because it's one of my favourite ones. But this was taken in Horsham, near Horsham in Western Victoria, in a very dry year. It's not my photograph, but the person who took it has given it to me to use. And that was 2 separate paddocks, and where you see that green strip, that's where the fence was. And all the farmer has done is taken out the fence and just combined 2 paddocks into one, so he's sown a wheat crop across the whole lot, which was originally 2 separate paddocks. Now, the history of those 2 paddocks on either side of that, where the fence was that that's been in wheat production for 30 years. It's been no-till for 30 years. It was a grassland, was sprayed down and cropped, direct drill. It's never ever been cultivated, but it's had massive amounts of nitrogen fertiliser put on it for the last 30 years. In this very dry year, when he planted the whole lot to wheat, the only place that the wheat has grown is on the fence line where it hasn't been fertilised for the last 30 years. So the microbes in that soil where it hasn't been bashed and nuked and whatever with fertiliser have actually allowed that wheat to grow. So my question is, we talk about droughts and we talk about reduced plant production in a dry year. Did we do that? Have we done that to the soil? I mean, yes, the rainfall was below average, well and truly below average, but if he hadn't been using all that N, theoretically that soil is still capable of growing that crop. And the microbes around those roots are still capable, even though it's it is a monoculture, but the other thing was too, along the fence there would have been weeds. So for the 30 years where he had monoculture, he had a monoculture that was fertilised with N on both sides of that fence. And both of those things are really bad for the soil. Monoculture is bad for the soil, the fertilisers is bad. So along the fence there was no fertiliser, and there was also just a diversity of weeds, which obviously has just kept the soil microbiome alive, and I think that picture really does tell 1,000 words. So just briefly then, nitrogen. I don't have anything good to say about this substance. It only 10 to 40% is ever taken up by the crop in the year of application due to substantial losses. We get denitrification and volatilisation, leaching, surface runoff. Like such a mobile element, it's going to go somewhere else. Plants can't possibly take up all the N that you put on them in one go. I mean, the problem is, you imagine if you had a day-old chicken and it's just newly hatched out, and you said, oh, here you go, here's your feed for the next 12 months, and just put it there next to you and, go for it. You've got all the food that you need for the next 12 months. Well, I mean, a day-old chicken, it can't possibly eat all that food, and over time it's going to deteriorate, isn't it? It's not going to be still good quality. I mean, a chicken needs to be fed every day. Plants need to be fed every day. So we front load them with all this stuff and expect that a new seedling or a new seed can possibly use all that, or even if we're putting it on incrementally, we're still putting on way too much, and a plant cannot use it all in that amount of time. And the thing is that because you've put that onto the plant, you've also interfered with the communication between the microbes. So if we just look at this in terms of just financially, globally, farmers spend $100 billion on nitrogen fertilisers, and if 60 to 90% of it isn't taken up by plants, then that means there's $60 billion to $90 billion either goes up into the atmosphere or out into the rivers and streams, or out into the ocean and pollutes the water. So it's massive. Just the financial side of it doesn't make any sense at all. And of course the environmental side, you're all well aware of it up here in the reef area, but there are 500 oceanic dead zones around the world due to fertiliser runoff, and some of them are probably a lot worse off than the Great Barrier Reef is. There's some waters that are totally eutrophied. I mean, you think of that area in the Gulf of Mexico, that dead zone, I can't remember what it is, the size of, so it's big anyway, it's huge. And then, Chesapeake Bay and everything off the northeast coast, then you've got the Baltic Sea, which is just incredibly eutrophied. I mean, all of these things are due to fertiliser runoff from farms. So farmers are losing out, farmers are spending all this money, and then the fertilisers are causing off-site impacts that we don't want for our greater environment. Inorganic nitrogen, and inorganic N, I'm talking about things like anhydrous and urea and ammonium nitrate or calcium ammonium nitrate, any of those kinds of things. Inorganic nitrogen forms non-protein nitrogen in our plants, which is not healthy for our plants, and they're going to attract insect pests. Very unhealthy for any animals that eat those plants, incredibly unhealthy because they can't digest non-protein nitrogen, and it stimulates weeds. So again, we're just causing more and more and more problems. So they're expensive, inefficient, polluting. Why on earth do we use them? Like, where did this idea come from that we would use this stuff that's not of any benefit to anybody other than the person selling it? And of course, everything that you see, I just downloaded this from the internet the other day. was just a little thing for kids. It's WikiGrow, like how to grow flowers from seed. And it says, and just, plant your seeds and apply fertiliser. I mean, isn't that just like standard, even right through to, even if you buy a packet of seed from somewhere, it'll tell you how to grow something. It'll say, to dig a hole for your plant and apply complete fertiliser and put your plant in. Like, it's just so ingrained in our thinking that a plant can't grow without fertiliser. It's incredible. It's very, very hard to change that, actually. And I remember when I was teaching in the Department of Agronomy and Soil Science at the University of New England, and someone told me that dairy farmers on the north coast were starting to move away from nitrogen fertiliser because it's having such a detrimental impact on their cows. Was increasing mastitis and laminitis, and they're having liver dysfunction problems and all sorts of things. I thought, well, how could nitrogen fertiliser do that? Like, really? I didn't believe it. And secondly, how could you grow grass without nitrogen? I mean, if you had to grow all that grass that a dairy cow needs, you need really high producing pastures, like how could they possibly do that without nitrogen? So I had been so brainwashed about the fact that plants won't grow without nitrogen and what's wrong with nitrogen. Like people will still say, scientists will still tell you that the nitrogen you buy in a bag is exactly the same as the nitrogen that would be fixed in the soil. So, they'll tell you that there's no difference. So, we still have a long way to go with educating people about fertiliser. And it's true that fertiliser does make plants grow. That is true. Apply N and they will grow more. You will get more biomass. But are they just empty vessels? The thing about the extra growth is there anything else in it? And if you're looking at it from a livestock point of view, there's really good information coming out from the dairy industry that shows that, yes, you do grow more grass. There's lots and lots of science. You can use a rising plate meter or something more sophisticated than that. And if you add nitrogen to a pasture, it will grow more. All that happens is that the cells in the plants elongate, and so it's taller. But now that it doesn't have all those exudates coming out of the roots and it doesn't have communication with the microbes in the soil anymore, it hasn't got the zinc and the copper and the cobalt and all the other trace elements and things in it that it needs, so that the animals that eat it are not getting the nutrition that they need and they're not able to produce any more milk. So there is actually no relationship between the amount of nitrogen applied and the amount of milk produced in a dairy. And this is what it looks like. This was put together from the... what are they called? Down in Victoria anyway, but it's the Milk Research Farm, Ellenbank Research Station in Victoria, where they do all this research on dairy pastures. And they just looked at milk production over these years here, and the reason that milk production had increased, that's that red line, over the time of that study, was because of improved genetics in the cows. They actually had higher milk producing cows. The black line is the amount of nitrogen that was applied over that time, and this is for the all Australian milk production. This is not just for Ellenbank Research Station. This is for all milk produced in Victoria, South Australia and Western Australia over those years. There was no relationship whatsoever between how much nitrogen farmers used and how much milk produced, because there isn't. And I've talked to, like, even in New Zealand where dairy is really big, and I've gone to the research stations there and I said, show me the relationship between nitrogen applied and milk production. They will show me the relationship between nitrogen applied and grass growth. And I said, but that increased grass growth is not increasing milk production. They cannot produce the data. So the scenario for grains and vegetables is similar, but the lack of nutrient density, in other words, we've just got empty calories. You might grow more wheat or something, but it's not going to have anything in it of any value. It goes largely unrecognised. So Tegan was talking about, the nutrient density meter, and that's the kind of thing that we need, like farmers need to be paid for the nutrient density of their product rather than the weight of it, because once they start getting paid for nutrient density, then we'll see some changes, hopefully, in the way farming is conducted. So if we look at phosphorus, it's pretty similar to the nitrogen story. If anything, it's worse. Only 10 to 15% of applied phosphorus is taken up by plants in the year of application. Again, that's the science, that's what's published. It's not my opinion. So in other words, 80 to 90% of it's fixed in the soil. So phosphorus, while nitrogen is very mobile, phosphorus is very reactive. And it's just going to react with whatever, cations are there. It's a negatively charged anion. and it's going to link up with something positively charged like iron or aluminium or calcium, depending on the pH of the soil. If the soil's pH is less than 7, what is the pH of these soils around here? Are they acid? Acid soils? 6? All below 7, are they? Pretty much. So it's going to just tie up with aluminium or iron or manganese or something like that in an acid soil if the pH is below 7. If it's above 7, like if you go inland, I'm sure you'll find pH like alkaline soils inland, then it's going to link up with calcium and cause insoluble calcium triphosphate. But again, you know, when we look at soil tests for phosphorus, all they're telling you is about available phosphorus. In other words, what's in solution, what's really readily available. And phosphorus it binds with things. It's not in solution. It's not readily available. So when you look at this figure here, which I know is a bit complicated and I don't want to go into it in any detail, but the available phosphorus is that bit over there on the right-hand side. It's what's actually, like I said, in solution. It's about 1 to 3% of the total amount of phosphorus that you've got in your soil. So you could take your available phosphorus level and multiply it by 100, and that's going to give you something more like how much you have there in total. And in fact, some, like in New Zealand, they actually tell you that on the soil test report. They'll tell you that your Olsen P level, which is the extraction method that they use for measuring phosphorus in New Zealand, tells you what percentage it is of your total P. They probably should start doing that in Australia as well, telling you that really the other 99% or 97% is still in your soil and not being measured. The other thing about phosphorus is if you apply it as a fertiliser, it's immobile, even though it's reactive. It doesn't move. So it's not going to move more than 4 to 5 centimetres. In other words, like more than 2 inches from wherever you put it. So if you're in a situation where you've got plant roots down, 20, 30, 40, hopefully 50 centimetres or a metre down, what on earth is the point of putting phosphorus, in the top little bit of soil? There's no way that the plant roots can get it. Only a newly emerging seed can get it, or a seedling can get it, and that's when you do not want that plant to have access to phosphorus, because that's going to shut off its mechanisms for forming relationships with mycorrhizal fungi. Oh, we've got a little message have come up here that doesn't matter, I suppose. It just says we've got a problem, but it's not showing up on there, so we'll just ignore it. We'll just ignore it. So phosphorus fertiliser is incredibly inefficient, and I'll bet farmers are not told about that. But it's also incredibly inhibitory because it prevents plant roots from sending out a hormone called strigolactone, which is the hormone that a plant root produces to tell the spores of or to signal to the spores of mycorrhizal fungi in the soil to germinate and to grow towards the plant root. So they will, when spores of mycorrhizal fungi detect that strigolactone in the soil, they then use that concentration gradient to find their way to a plant root. Because again, if you think about it, a spore of a fungus is a tiny, tiny little thing. And if it germinates and grows off in the wrong direction, it's not going to find a plant root in time. It actually has to form a relationship with that plant root to get its energy. So it's only got whatever energy is in that tiny little spore. That microscopic spore is just enough energy to get to a plant root. So it has to follow that concentration gradient of strigolactone. Plants will not produce strigolactone if you apply phosphorus to the soil. So you just muck up all that conversation. They're vital to root formation as well. And as I said, colonisation by mycorrhizal fungi. So if we don't have that colonisation with mycorrhizal fungi, then the plant is going to be deficient in the micronutrients that the fungus can bring back, such as copper, zinc and selenium, and then farmers are in the situation of having to apply those things to plants. Or if you've got, like the question you asked before about feeding supplements to animals, if those things are not in the plants, the minerals aren't in the plants, then the animals can't get them either. So it's usually a soil function problem. If we're having to give supplements, then there's something going on in the soil. And in fact, for phosphorus anyway, if your soils are really healthy, you won't get a response to added phosphorus. So you could put test strips out and nothing will respond to it. But this reduced uptake of minerals is a real problem with using phosphorus because it has negative flow-on effects for animal health and also for human health, because we're either going to eat those plants or we're going to eat the animals that ate those plants. And then the plants themselves also suffer from having the mineral depletion. So it's not just the things that eat the plants, but the plants themselves, they can't defend themselves against pests and diseases because they need to have zinc and copper and cobalt and whatever in their bodies to for their immune systems to work. And then the farmer is put in the situation of having to use insecticides and fungicides. And as I said before, if you've got insect attack or fungal attack, you're going to have to use something like that to save your crop. But you need to think about, well, how could I do things differently in the future so I'm not putting myself in that position where plants... and Tegan also mentioned using a refractometer to measure bricks. We know for sure that if you can get your bricks over 12, that you're not going to have problems with insects. It depends on whether they're sucking insects or chewing insects, and sucking insects definitely are inhibited by high bricks levels. So things like aphids, for example, you're always going to find they're on low bricks plants or on low bricks parts of the plant. And then chewing insects would be things like grasshoppers and caterpillars and stuff. And you need slightly higher bricks to inhibit those, but for sure, if you can get over 12 or 14, you won't see chewing insects on your plants either. I've seen lots of examples. If you look at plants that are, like say some of the like forage brassicas and those sorts of things, the lower leaves that have got lower bricks because they've transferred the energy to the more newly emerged leaves, you'll see the lower leaves have got holes in them, and then the new leaves that are higher bricks haven't got holes in them. So you can see where the insects are actually attacking the low bricks leaves, even on just the one plant. But certainly you'll see big differences between crops, and you'll see crops next to each other, one high bricks and one low bricks. The low bricks one will be full of chewing and sucking insects, and the other one will be fine. So bricks is really, really important for protecting the plant against insects and fungicides. And what's high bricks? High bricks is high levels of sugars, but also very high levels of minerals. If you use your refractometer, how many of you got a refractometer? Have you used a refractometer? Oh my goodness, I want to see 100% of hands go up. Right, well, get yourself one. They're only about $100. Make sure it's got a nice clear gauge or whatever you call it on it, so you can see the numbers really easily. And don't just buy it, use it, and use it a lot. You know, go out and start measuring plants all over the place, on the sides of the road, different times of the day, different kinds of plants, different leaves on the plant. So if you've got a plant, you know, that's this high, measure some of the top leaves and the middle leaves and the bottom leaves and figure it out for yourself, what is actually going on in those plants. Measure them in the morning and measure them again in the afternoon. If it's the same in the morning and the afternoon, you're in trouble. Because if it's the same in the morning, it's what it should actually be increasing during the day, because you're measuring photosynthesis. The sugar levels in the plant should increase during the day. And if they're not, then it means that all the sugar that was formed during that day was not downloaded overnight, and it did not go down into the roots and didn't go into the soil. So if you might, some people say, well, we've got really high bricks levels, but we're still getting insect attack. And I go, well, go measure in the morning and go measure in the afternoon. And I say, well, yeah, it was 15 at 9 o'clock in the morning. And I go, well, you've got a problem, because that plant did not download its sugars overnight, and that means it didn't get, it's got no contact with the microbes around the roots for some reason. Maybe there's insecticide or fungicide or something in the soil, so there's no communication there, so it's not taking up minerals, and that's why it's getting attacked by insects. So all these things that farmers are using are costing lots of money, as I'm sure everybody in this room is aware of. So this data is from Canada, but it applies equally well to Australia. So this is Canadian net farm income over an 80-year period from 1926 to 2016. And the blue line is the gross value of agricultural revenue. And over that period, the blue line has been going up. And it goes up and down, as we're all aware, with commodity prices are always fluctuating, seasons are fluctuating, so you expect that gross revenue is going to fluctuate. The green line is net farm income. For a certain amount of time on that graph, up to around about, 1950s or something, you'll see that the green line and the blue line were following each other very closely. In other words, when gross income is higher, then net farm income will be higher. And that's what we'd expect in a good season. When the value of agricultural production is higher, or when commodity prices are high and the value of agricultural production is high, farmers should earn more, right? And in a poor season, well, you know it's not going to be so good in terms of cash flow in a poor season. But then all of a sudden, the blue line, it keeps on going up, and the green line, net farm income, goes down. In fact, it's gone into the red there in 2013 2006 was when in Canada and North America as well, the cost of corn was higher than the value that farmers received for it. In other words, the cost of production of corn was higher than the revenue received for growing corn. And that happens with lots of commodities, as those of you who sell into international commodity markets are aware, that you have those negative income years. But that blue area, man, that just kept on going up. I mean, look how that blue line is going. Well, who's making that? Someone's making a lot of money out of agriculture, and the guy who put those graphs together, Darren Corman, he actually published a book on this last year as well. He found that in the last 32-year period in that graph from 1985 to 2016, input suppliers, they're the guys that are selling you the seed and the fertiliser and the insecticide and the fungicide and everything, they received 1.32 trillion of the gross revenue of 1.35 trillion, in other words, 98%. So 98% of gross agricultural production went to input providers. And when someone talks to me about, well, you know, don't you think I should add this or add that or supplement my animals with this, that or the other, I go, oh, there's the 98%. Someone is selling you something. And the input providers are doing very well, thank you very much. Luckily, Canadian farmers, or luckily for them, I suppose, they have taxpayer-funded support schemes, as they do in North America and much of Europe. You know, the basic payment in Europe is incredible. Like, you know, a farmer can get $90,000 a year for just sitting there, really, and saying, I'm a farmer. And, you know, and the money just comes to the mailbox, provided you've got a couple of animals and you do some things. You'll get paid, whereas we don't have that here in Australia, so we have to produce some stuff. And but in fact, I think probably we are better off in the long run, because I don't think those support schemes really are beneficial. But anyway, farm debt in Canada, it's at a record high, around $100 billion, and farm debt in Australia is also at a record high. I've got the figures on that, but I'm sure that you don't need to see those. You will know that farm debt is high. And we're told that we have to produce more to get out of this mess. That's always the story that we hear, get big or get out, or, you know, that you need to increase your yield. All this stuff we hear about yield, yield, yield. It's got absolutely nothing to do with maximising yield. It's actually all about optimising profit. And there's not enough emphasis given to that. And we have to regenerate the resource base. So if we look worldwide, we see that this excessive use of nitrogen and phosphorus fertiliser has caused soil degradation. There's lots of science on that. It has caused environmental pollution. I'm sure you all hear about that and are aware of that. It has reduced soil biodiversity. There's lots of science on that. And it has caused trace element deficiencies in plants, animals and people. We have lots of evidence on that as well. So that best management practice, if you like, that we've been using for the last 50 years or so quite clearly has failed. It's been a big fail, really. And now that, as I said, we're seeing, you know, massive infertility problems in the human population and massive health problems in the human population, perhaps we should take note that it is time we need to change. So let's talk about supporting these microbes, these amazing things that work so hard in the soils and plants and animals, rather than using high analysis fertiliser. And as I said this morning, we've got several options for that. We could use a biostimulant, which basically means to stimulate biology. A biostimulant theoretically shouldn't have any nitrogen or phosphorus in it, or very little. We are not trying to apply any kind of fertiliser to plants. We are just wanting to stimulate microbes. That's my definition anyway of a biostimulant. We can look at plant diversity, and I talked about that before lunch. That is something that has taken the world by storm, really. Multi-species everything is going to become the norm, thank heavens. And we can also look at biostimulant and plant diversity, what happens if we put those things together. So a biostimulant is just really something that stimulates or supports the soil microbiome. And I get a little bit sort of, I can't quite understand it sometimes when people tell me that they're using a biostimulant but they're still using synthetic fertiliser, or they're still buying seed that's got insecticide and fungicide and everything on it, and then they put biostimulant on it as well. And I figure it's probably a little bit like taking a probiotic and taking, you know, antibiotics at the same time or something. I go, just don't you really quite get this? that if you're going to use a biostimulant, you have to wean off those other things that you're using. You have to figure out how to reduce those. Maybe not going cold turkey, but you definitely have to reduce them. So there's lots and lots of biostimulants on the market. There's lots of biostimulants that you can make yourself. There's lots of very cheap and simple things that you can use as a biostimulant. For example, milk works perfectly as a biostimulant. That's all right. I can send information to Julia, if you like, about the use of milk, and other things. But so it doesn't have to be expensive. So what you're trying to do is, just really nurture the microbes that are already there. So remember, a plant has a core microbiome. A seed has a core microbiome. You do not need to add microbes. There will be microbes already there. But what we do want to, or what we could do to stimulate the life in the soil is apply autoinducers. So when I talked about quorum sensing, I said that autoinducers are the chemical signals that microbes use to communicate with each other. And in microbially rich environments, like in a fermentation environment, for example, like in the rumen of a cow, I said 1 drop of rumen fluid has 10,000 times more microbes in it than there are humans on the planet. So that's a very, very microbially rich environment. There is going to be a lot of chemical signalling going on in that environment just because there's so many microbes in there and they're all talking to each other. So another chemically or microbially rich environment is in the gut of an earthworm. So an earthworm's ingesting material and then it's undergoing anaerobic fermentation, which is the same as what happens in the rumen of a cow, anaerobic fermentation. And so vermicast or vermiliquid or anything that an earthworm produces also has a lot of autoinducers in it. In other words, it has the chemical signals that the microbes used when they were communicating with each other. So what if we take those materials that have a lot of autoinducers in them and put them on a seed? What happens when you put them on a seed? Now just imagine if you were a seed. Seeds are pretty smart too. There is plant intelligence as well as microbial intelligence. If you take a seed and plant it 10 metres down or 10 feet down even, it's not going to germinate. It's not silly. It knows it's nowhere near the soil surface. So if you could leave it there for a couple of years even, and then you could bring it up and you could plant it at an appropriate depth near the soil surface and water it, and if it was the right time of the year, it would come up. So how did it know that now it's 1 inch below the soil surface rather than 10 feet below the soil surface? How does it know that you watered it? How does it know what time of the year it is? If you go to seasonal rainfall environments, like right up north or, down to Victoria or something where it's, it's either going to be like a wet winter or a wet summer, plants know. Like a plant is not going to germinate at the beginning of the dry season. So how does it know there's a dry season coming? You think about that. When you think about the native seeds, the native plants, they come up at the right time of the year. How does a seed that can't see, can't think, can't hear, can't talk to other seeds, doesn't look at the weather forecast? Mind you, what is the weather forecast? They keep telling us down there in New South Wales that we're going to get a 90% chance of 20 to 40 millimetres next week, and that always disappears before next week gets there. But the ants know. I take notice of the ants. They know when it's going to rain. But a seed knows as well. And a seed actually knows probably a lot more than we do. And a seed knows. How does a seed know? Well, a seed senses. It senses through its seed coat. So it's sitting there in the soil. Just close your eyes and imagine I'm a seed sitting in the soil and it's wet out there. It's moist. Okay, so that's going to come through my seed coat, right? I'm going to feel, it's moist out there. And it's going to detect the temperature, and it knows how far from the soil surface it is by the infrared, or not the infrared, the wavelength of the light that's coming through. It knows what plants are growing around it because it picks up all the chemical signals that are coming from those plants, and it knows what microbes are around. And it needs to know what microbes are around because that's really important for a seed, because it's got to form a relationship with those microbes to grow. So let's say that we take some vermi liquid, which is a really good example because any of you can make your own vermi-liquid. You don't have to buy that. So you'd have a worm farm and you collect some worm juice, right? And you can just put that on a seed that doesn't have insecticide and fungicide on it, and you're not going to put any high analysis fertiliser with it. So if we compare a seed that had that worm juice on it to one that doesn't have the worm juice on it, the one that does have it, what's going to be in that worm juice? Lots of autoinducers. Thank you. You have been listening. And what is the seed going to think when it imbibes water and all those chemical signals come through the seed coat? Seed is going to sense, hey, there's a whole lot of microbes out there because I'm picking up the chemical signals of a whole lot of microbes. You put them on the seed, but it doesn't know that. That's one thing it doesn't know. So it's important for that seed to form a relationship with those microbes. So there's lots of exudates are going to come out of the seed, and also the new roots of that plant are going to produce massive amounts of exudate to feed the microbes that it senses are there. They're not there, but we've tricked it into thinking they are there. So it responds as though they are there, feeds them, and what it does is then feed the resident microbes that are already in the soil, and they proliferate, form a relationship with the plant. And because the plant's producing lots of exudates for them, we've tricked it into producing lots of exudates, then they're well fed, so they bring lots of nutrients to the plants, and we just kickstart that whole relationship between the seed and the soil. And in my way of thinking, that's what a biostimulant needs to do. We are not adding microbes to a seed. We are adding the signature of microbes. The chemical signalling molecules that microbes use to communicate with each other is what we are putting on there. So I mentioned this morning Derek Axton. Derek and Tanner Saxton in Saskatchewan in Canada, they grow polycrops. So they put all of their crops are polycrops, or nearly all their crops are polycrops. And they've got one of these big optical seed graders, and they'll plant a whole heap of things together and separate the seed. But they also make their own vermi-liquid, and they're farming 6,000 acres. Acres, I think. Yeah, they still talk acres up there. And doing it fairly cheaply, making plenty of money, and they can pay for millions of dollars worth of optical seed grading equipment. But this is just a photo of, this is some Durham wheat. That's I just took this photo this year on Derek Axton's farm. So the seed is just there. For those of you who are looking on the other screen, it's like an inch or so below the crown of that plant. And then you've got that amazing root system coming down out of the seed, all that. Like, this plant is very, very young. It's only got like about 2 or 3 leaves, and it's produced a wonderful seminal root system, like the roots that come out of the seed, and very deep, and you'll see there's rhizoschiests all the way down, and that's going into a very heavy clay soil. So people will say, oh, rhizoschiests will only form in sand, you won't see them in clay. That's not true. You actually will see them in clay. And then you'll see there's some roots coming out from the crown, which is like at the roots, at the soil surface. There's like 3 main roots coming out from there. And they've all got great rhizoschiests on them as well. So we've got really, really good root development. You would never see anything like that in such an early stage of growth of that plant if you'd put high analysis fertiliser under that seed or near that seed, or insecticides or fungicides on that seed. And then this next photo is also from Derek Axton's, and these are oats, and these are more mature plants, and you can see that there's great rhizoschiests on those plant roots. So that's putting a biostimulant on the seed, and he makes his own, so it doesn't cost him a lot. The Haggertys that some of you may have heard of in Western Australia, Ian and Diane Haggerty, they put vermi-liquid on their seeds, on their wheat seeds as well, and they also use compost extract, not compost tea, but just extract. So the thing about compost extract is that it is it's just a cold water extract of compost. So you're not taking the microbes out, you're just taking the autoinducers out. So when you use compost extract, you're putting autoinducers on the seed, and tricking the plant basically into thinking that it's in a microbially rich environment when in fact it's not. So compost extract is going to be similar to vermi-liquid, probably not as good, but if you make a fermented compost, and I think you might have had people up here in Queensland actually, in the dry tropics anyway, there's been David Hardwick and others talking actually about how to make fermented compost. It's very easy to do, and that in that fermentation medium is going to produce a lot more autoinducers than aerobic compost. So any kind of a fermented anaerobic environment actually people go, oh, you can't make compost, it can't be anaerobic. Well, it can be anaerobic if it's been inoculated. You make an inoculant and you get the lactobacillus working in there, and it works extremely well. So all the best systems really are anaerobic ones. And when you think about it, humans have used fermentation for thousands of years to preserve food, and it's a very popular technique. We've understood about fermentation for a long time. So that's there's lots and lots of different kinds of biostimulants that you can use, and if you're interested in that area, talk to each other, look at the literature on it and everything. So biodiversity also supports the soil microbiome, and again, I talked about that this morning. It's going to be effective when rates of high analysis fertilisers are reduced. Again, I can't really see the point in putting in a 12-way mix or something and sticking high analysis fertiliser under it. You're defeating the purpose. And most people now that I'm aware of don't actually use fertilisers with any of their multi-species mixes. So this is an organic farmer in Manitoba, again in Canada. So this is just a cover to, this is what it looks like just in the very early stages of growth. And all he's trying to do there is, this is just something that he puts in between his cash crops just to get his soil working again. So there's no fertiliser used. He's got faba beans, chickling vetch, oats, cereal, rye, field peas, sunflowers, buckwheat, sorghum, kale, forage brassica, phacelia, tillage radish and brown mustard, and no fertiliser. So he's having a great effect on his soil. These are just some plants that we pulled out. By the time I took the photos, they'd wilted pretty severely, but there's faba beans and oats and chickling vetch and something else and barley there. You can see they've all got riser sheaths on them, the whole lot. So this morning I showed you some screenshots from the Yana video, just looking at, what's happening around plant roots. And what we know is that with that life that's around plant roots, that the nitrogen actually, some of those bacteria that are around plant roots need to be free living nitrogen fixing bacteria, and the nitrogen actually has to be fixed by them biologically in order for stable carbon to be formed. Because the stable carbon molecule, when we actually look at that molecule, this is a diagram of the humus molecule. This is the holy grail. This is what we want to have formed in soil. You see that there's carbon and oxygen and nitrogen and hydrogen all in that molecule. And all of those atoms of all those different elements have been joined together into that polymer by microbes. Microbes have polymerised all those elements. Microbes have formed those bonds between all those carbon, hydrogen, oxygen and nitrogen atoms and formed that really incredible, stable, humic molecule in the soil. Why on earth would they bother doing that? Just think of all the energy that went into doing that. So you're a microbe living around some plant roots, one of these ones here. You're just, in that lovely world with all these exudates coming out, and you could just feed on all those sugars and breed and, just have a great life just being a microbe and reproducing, right? Sounds pretty good. But then, no, you have to work. We're actually going to use some of that energy to stick all these elements together and make humus. Why should we bother doing that? We could just be, eat, drink and be merry kind of thing. So why are microbes putting all that energy into forming humic molecules around plant roots? Why would they bother? Yeah, to create a home. So they're going to form riser sheaths around the roots. So that's going to be a much more protected environment for them, and it's going to be a much better environment for the plant too, isn't it? So if there's a riser sheath around a plant root, then it's really hard for pathogens to get in. Like, how are fungal pathogens going to get in through that for a start? It's like building a wall around a castle or something. But what else? So remember, look at it from a microbial point of view. You could just... sorry? A better future. Well, exactly. Absolutely perfect. Well, if you've created a better environment for the plant to live in, so humus has got a high cation exchange capacity, high water holding capacity, you've got better soil structure, your host plant is going to survive better, right? That means they're going to keep on feeding you. Because if you're a microbe, you're going to die when the plant dies. So you've got to keep that plant alive, and we interfere with that. Yeah?
Just make, say, a more resilient home for itself.
Absolutely. So it is in its own self-interest They're acting in their own self-interest to keep that plant alive. And we interfere with that by sticking things around the seed. All right, I think I've made the point. All right, so phosphorus solubilizing bacteria, which we also need living around plant roots. And we're going to eliminate those if we put any phosphorus near the root, near the seed. They stimulate the biological nitrogen fixes. So when you read the literature about what happens in the rhizosphere, you'll see all this stuff about PSBs, phosphorus solubilizing bacterium, BNFs. It gets very complicated, but if we add phosphorus to soil, we actually inhibit the entire soil building process, including stabilisation of carbon, because we need nitrogen to be fixed biologically to go into that humic molecule, and the nitrogen can't be fixed biologically unless the phosphorus solubilizing bacteria are there to stimulate the biological nitrogen fixes. I'm confusing myself now, but I think I got that right. Point is, what happens down there is very complicated, right? And just get out of the way and let it get on. The microbes know what to do. So what happens if we use a biostimulant and biodiversity? So we're going to not use any high analysis fertilisers at all. We're going to have a very diverse group of plants, and we're going to stimulate them with some kind of a biostimulant. How am I going for time? Yeah, okay. So just one example. Those of you who've seen my recent YouTube videos, you've probably seen this one before, but it really blew me away. It was the best example of rapid soil building that I've ever seen in my entire life, and I'm still have not seen anything better than this one. So this was Jenny and Maya Smith, Autoinducers in New Zealand. It's near Rotorua. And they're on a pumice soil, which is an ash soil, volcanic soil, very, very high in silica and very low in just about everything else. So it's a bit like beach sand, really, except that it is a volcanic soil. If you send it off to the lab to get tested, it'll say it's got no nitrogen, no phosphorus, and no zinc, boron, copper, cobalt, selenium, anything. So farmers are told that they need to add everything that plants need and animals need in order for these soils to be productive. That's a close-up view of it. So Jenny and Maya were spending a lot of money. They're dairy farmers. They were spending a lot of money on fertilisers. Then they had all these pest problems. They had grubs that come and eat the roots of the plants, and health issues with their livestock, reproductive problems in their cows, like 20% empty rates, and high somatic cell counts in their milk, etc., etc., etc. So Maya came to one of my workshops about 3 years ago in New Zealand, and I was talking about all the issues with nitrogen, and he stopped using nitrogen fertiliser. They weaned off nitrogen, and he was very pleased with himself, and he showed me this photo of the topsoil that they'd built, weaning off nitrogen. That's his son Taylor that's holding that. And you can just see in the background there that it's a ryegrass and clover pasture. It's a reasonably good pasture. So I'm just going to turn Taylor upside down for a minute if he doesn't mind, just so you can see the soil as we would see it. So there's about like 6 inches of topsoil there that they've built from getting off the nitrogen fertiliser, and he was very, very pleased with that. And I said, well, Maya, 3 years ago I didn't really know what I know now about diversity, and you could really improve that a lot from going from ryegrass clover pasture through to a diverse mix. And he goes, oh, geez, I thought we'd made it, like we've actually built some topsoil. I said, well, I think you could build a lot more. So he just tried a little, just a 2-acre paddock. He picked the worst part of the property, and they put this field of multi-species mix in. And you can see those cars down there in the corner. That's sort of like where the gate is into the paddock, and then there's a fence comes up from there and another one along the road there. The water tank is in the paddock and the sheepdog is in the paddock. So it's just a tiny little trial area of a multi-species mix. And he had 12 different things in there, and we went in to have a look to see whether the red clover had inoculated. And so that's Maya with the spade, his wife Jenny next to him, and Taylor, their son, and then that's Claire Bradley from Agrasea. Agrasea is a company in New Zealand that produces a fermented seaweed product. And again, I think it's very important that this is an anaerobic fermentation process. They just basically chop its kelp, they cut it up, they put it in big tanks, and they just sit it there like for 6 months basically, and it just ferments. So this is just the liquid. It's like a seaweed extract that comes from that. It's got nothing else added to it. If you analyse, it's got virtually no nitrogen, no phosphorus or anything in it, but it's got absolutely would the amount of microbes involved in that fermentation process will be huge, so it's loaded with autoinducers. So that's gone onto this field as well. And when Maya dug the soil up, this is what he saw. So this is what it was, and then it was that. And he didn't react at all. He just looked at it and he went, oh, we must have burned a tree here. Because, I mean, how could you, I mean, really, you know. It was like, so he just put that back. And then he went over here and he dug another hole and it came up exactly the same as that one. Actually, in that photo, his son Taylor is pointing to, there's a little tiny bit of pumice right at the bottom of that hole. But the rest of it, he's got like, 30 centimetres of beautiful, rich, amazing topsoil. And it was full of earthworms as well. He said, we've never ever seen an earthworm on this place. So we kept buying them, truckloads of earthworms, kept putting them on the farm. And then, then we could never find them again. So someone said, oh, you need to put compost on for the earthworms. So we bought tons and tons of compost and we put that on and earthworms. He said, then we never found the earthworms. And then, we just planted this multi-species pasture. And if I can press the right things here, now we've got all these earthworms. So anyway, so he dug a hole there and it was the same. And then he went further away and dug another hole and it was the same. And then he went like about 10 metres away and dug a hole and it was the same. And it was like, oh my God, I really have got all this topsoil. Like, it's for real. I've got a whole paddock of this. And he just went crazy, absolutely crazy. Dug 50 holes in really short amount of time. And he wasn't even putting the soil back. There was like soil going everywhere. I wish I had it on video. It would have been like truly a spectacular video. This poor guy was just, I have actually built all this topsoil. He said, and then he was almost in tears. He said, this has been like a dream. All my life we wanted to turn this pumice into something. And when they did that, they thought they'd done a fantastic job. So that's and the reason that I'm so impressed, I have seen soil building before on other places, but that multi-species crop has been there for 5 months. That's what really blew me away. Now, they had been off nitrogen for 3 years and using a biostimulant for 3 years prior to doing this. So whether you went straight from, using lots of nitrogen and having a low diversity pasture, whether you'd get that in such a short amount of time, I don't know. But that's why I say it's the most rapid example I have ever seen of soil building, and it happened in such a short amount of time. So that's from that pasture looking out now to the rest of their farm, which is all ryegrass. And in the background you can see some hills which are totally bare. That's Crown land owned by the government, and it was covered in pine trees, which a government plantation of pine trees, and they have clear felled all the pine trees on that hill. Look at the steepness of that country. That is absolutely, if I can find the red pointer here, absolutely amazing. Look at these hills. They have been planted of pine trees and then clear felled of pine trees. Just think of the erosion that's going to happen there. And what is the New Zealand government doing about climate change and emissions? They say, we're going to plant more pine trees. Great. Will they get funding for this? No. Well, you can't build soil carbon. Why do they think you can't build soil carbon? Because the scientists do all the measurements on farms, research stations where they use fertilisers and they have monocultures, and you're not going to be able to do it with monocultures and fertilisers. So the plan now is for these other fields here, which are all ryegrass at the moment, to be transformed to these multi-species mixes like this. And so that was May last year, and then just before Christmas, Maya sent me this photo of his legless cows, I call it. So he's also going to much longer rounds and letting it grow. Again, with dairy pastures, farmers will be told you've got to keep it short. They're letting it grow nice and long. Again, look at this hill and these mountains in the background. I mean, that's criminal, isn't it? But the government says that's fine. And so this is Jenny in their multi-species pasture when it's grown up. Isn't that an improvement on ryegrass? She's only short, but this is Maya in his chicory and other things. And this is quite close to their house, this paddock, and they say they'll just make a cup of tea and go and sit out there in amongst all the flowers and everything. They said that the butterflies and all these coloured beetles and all the birds and the life in that paddock is absolutely extraordinary. And to sit there in amongst all that life, their cows are doing better. Cows are breaking in actually to that paddock. They know where it is. And everything is doing so much better. Why would you just have grass? I mean, that's a huge paradigm shift for people. If you're graziers and you just have grass, just think about this, moving away from that. So the outcomes, the Agri have actually measured these, like taking soil tests. So soil tests are useful for things sometimes, but they found the cation exchange capacity has increased 50%. That's massive. All of the nutrients, including nitrogen and phosphorus, have increased, but none have been added in the last 3 years. Total organic carbon has doubled. That's huge in the top 12 inches of the soil. Brix levels have quadrupled, so they're only able to get Brix levels around about 4, which of course means they are going to be subject to insect attack all of the time because 4 is just way, way too low. So 16 takes them up over that magic number of 12, so they're fine now. They've got lots and lots of beneficial insects, which are all pollinators and predatory insects, but none of the leaf chewing insects or sap-sucking insects. Their milk production has increased by 300 litres when the cows come into that paddock. Somatic cell count, which is an indicator of mastitis and other things, is halved. Cow fertility has increased 60%. And Meyer has calculated that he's going to be able to double his stocking rate on that farm quite easily without overtaxing anything because he's just got, heaps of feed available now. And very happy cows. But very smart cows too, obviously, because they know where that field is and at night they're sneaking back there. So if we just look like in a global context, plant soils, microbiology, hydrology, all of these things are connected. We can't think about them in isolation. We live, the whole globe is a holobiome and everything is connected to everything else. And, since the Industrial Revolution, there's been massive changes around the planet, just in terms of how people have conducted the business of growing food, as well as a whole lot of other things. And we've really tried, we've simplified the landscape, and something we haven't probably really thought about. We've simplified our diets. Probably haven't really thought about that. Someone, actually when I was speaking up at Atherton a couple of months ago, someone from Poland, I think it was, came and said that back in like their traditional family farms, they had access to, the number of vegetables that they grew in their gardens, in their home gardens, was massive. A whole lot of vegetables that we wouldn't even recognise in, the names of them, the different sorts of things. And someone else on one of the islands, I think in the Philippines or somewhere like that, they said, there's over 100 kinds of vegetables that you can buy at the markets. You go to some of those markets in tropical countries and things and you look at all the huge, and think of all the different things that you could grow here, really, in this climate, in terms of fruits and vegetables, I mean. It's really crazy that we just focus on just a couple of things. We really need to think about diversity and, of course, diversity in the soil microbiome. So the fact that we haven't been thinking about diversity and we haven't really been thinking about soils as living systems, we haven't realised that we needed to feed the soil with exudates from a whole lot of different kinds of plants. So soil structure has deteriorated hugely. It's not just due to cultivation. It's due to a whole lot of other things. Cultivation has been blamed for a lot of things. Mostly soils that were cultivated were left bare. I think it was the fact they were left bare more so than the fact that they were cultivated. So this big switch that we've seen to no-till hasn't really improved soil health all that much, because what we needed was diversity. What we needed to do was to get away from bare ground. So if people have gone to no-till and they're just using glyphosate or other things to keep the soil bare, then there's no improvement in the soil health. So that leads to poor infiltration. And now what we see, we've got this drying landscape because a lot of our water is just, if it can't get in, it's going to evaporate, so it's going off as water vapour. And you can't see water vapour unless it condenses and forms into clouds, and it can't condense and form into clouds unless there are little things called aerosols which actually nucleate the water for it to be able to condense. And one of the main things that nucleates water vapour is bacterial aerosols that come from plants. So again, if we have bare ground, if it's either been cultivated to create bare ground or it's been sprayed out to create bare ground, or we've been mismanaging it and in dry seasons we don't have ground cover, like think about how much bare ground we have in a drought. If you go back to like prior to European colonisation, I was going to say, but there's other words I could use, but the invasion really of this country, I suppose, by Europeans. But if we went back to pre-then, in dry times there was still ground cover. There might have been dead grass, but there was still grass. There was still ground cover. We didn't have bare ground. Like, we have created massive amounts of bare ground in Australia by the way we mismanage in the droughts and by the way we manage our croplands. And we have dramatically changed how much water that should be in the soil has actually gone to the atmosphere. So we've got these poor infiltration rates, and Simon's going to be talking to us a little bit about infiltration tomorrow, which is a really, really important subject, because we need that water to actually be going into our soil, because it's important for productivity and it's important for the life in the soil. And I'm sure most of you will have seen these rainfall simulators. I don't know whether there's one here, Julia, somewhere. Do people use rainfall simulators to look at how much water actually goes into the soil? It's just a device that actually puts on, like an inch of rain or something. But what you'll see is that, we're obviously here, the one on the left with the good ground cover, with the grass ground cover, all the water infiltrated so that the container underneath that grass is filled up with relatively clean water, and the container at the front there has got no water in it. So there wasn't any runoff, the water ran in or infiltrated. And then right over here on the one on the right-hand side, where you just got bare ground, all the water ran off and took sediment with it. So, I mean, obviously, for the catchment health, for the health of the reef, we don't want all that sediment going into water, but it's a management thing. And again, this example here, just a fence separating, good ground cover on one side, poor ground cover on the other, and that's a grazing situation, that's not a cropping situation. You just see that water can't infiltrate if you don't have good structure, and you haven't got the structure because you haven't got the ground cover, and then that water is going to evaporate. And we know that water vapour is actually the greenhouse gas. Once it gets up there and it's a vapour, that is the greenhouse gas that has increased to the greatest extent since the Industrial Revolution, and there's plenty of science on that. It actually accounts for 95% of the greenhouse effect. So the figure there on your left-hand side, that's the contribution of water vapour to the greenhouse effect, and then the other greenhouse gases, carbon dioxide, methane, nitrous oxide, and then the other chlorinated fluorocarbons, whatever they're called, miscellaneous gases, shall we say. So, they're insignificant really compared to water vapour, and yet we're not putting enough attention on water vapour. And it just comes back to management and bare ground and not looking after the microbes in the soil. So we really need a quorum of people to start to realise these interconnections, how everything is interconnected, how we are actually influencing global climate through lots of different things that we're doing, including our land management. Then, when enough people get that, then I think we will be able to restore soil health and climatic stability. And as Tegan mentioned in her presentation, we need to really start looking at the nutrient density in our food as well, and being able to measure that, and maybe having consumers more aware of how their food is grown and what's in their food, and also the diversity, obviously, of things that they're eating. And don't forget, we need more flowers. So that's it for how much time have we got for questions?
Yeah, we do a quick question now. So don't hold back, be shy. We don't have much time, so last chance to ask Christine your questions or share some thoughts. There you go, Michael.
With your biostimulants, how does the rock minerals come into the system for cropping?
Okay, so if you your soil is comprised of rock minerals, as you know, and the top 50 centimetres of your soil probably has about 8,000 tonnes of rock minerals. The thing is that they're not available to plants, and if you apply like rock dust, then it's in a finely ground form, and it is easier for plants to actually access those minerals because of the form that it's very fine. But if you have good microbial activity in your soil, they will be able to access the 8,000 tonnes of rock minerals that you have.
So how do we get to transition to the good microbes?
Okay, so to transition to the good microbes, you need to think about, if you're planting anything, to maybe put a biostimulant that actually is rich in autoinducers onto the seed, to trick the seed into thinking there's lots of microbes there so it will feed them, and to think about plant diversity.
Yeah, but what about in sugarcane? We're talking mainly.
Oh, okay, all right. So if you're in a situation where it's hard to do those kinds of things, you could certainly use a biostimulant, for sure. And I would not be using microbial products. I would be using something that just stimulates biology.
You like molasses, seaweed, fish?
Yeah, fish would be great. Seaweed would be great. I wouldn't use too much molasses because that's going to stimulate bacteria in the soil that can burn up your carbon and have negative impacts on your soil structure. A little bit of molasses is okay, but don't go overboard with the molasses. Fish hydrolysate would be good.
Juice.
And
Chief.
what was the other thing you said?
Worm juice.
Yeah, vermi-liquid, worm juice, seaweed extract, fermented seaweed, fish hydrolysate, not fish emulsion, fish hydrolysate. All those things would be great. And if you're needing to add rock minerals, then you probably haven't got your biology stimulated enough. You have got 8,000 tonnes of minerals in the top 50 centimetres. And probably everything you need is there. But again, you know, if you can see a benefit from it, like if you try it and it works, well, it's obviously something that's going to be useful for you, and it certainly wouldn't be detrimental. Can't possibly do any harm. I mean, you can't possibly do any harm with doing something like that, but whether or not it's the benefit is, you'd have to assess that yourself. And everyone does have to, you have to kind of be your own agronomist, really. You have to start running trials on these things and seeing what does work.
You may have answered it a little bit already, but there was a lot of talk about agricultural soils being bacteria dominated, and I'm just wondering if we should be more aiming for a fungi, more balance, and can we stimulate the wrong microbes with our stimulants in our farming systems?
Okay, so good question. I didn't really mention bacterial to fungal ratios or fungi to bacterial ratios. We definitely want our soils to be fungally dominated. We want to have a lot more fungi to bacteria. Some people talk about a 1-to-1 ratio, but I've seen healthy soils that have up to like 15-to-1, 15 times more fungi than bacteria. So some of those fungi will be symbiotics, you know, things like, you know, mycorrhiza and stuff. But there's also a lot of really beneficial saprotrophic fungi that you can, and those saprotrophs will be there in those rhizomes sheaths as well. So if you're seeing those forming around plant roots, you know that there'll be lots of, it's actually fungal hyphae that pull those things together like that. Bacteria aren't capable of forming those kinds of aggregates in the soil. So that's one indication that things are fungally dominated. Also, if you have lots of beneficial fungi, you'll never have problems with pathogens around your roots because they'll keep away fungal pathogens. So yes, and definitely so things like fish hydrolysate and vermi-liquid will promote fungi. And what was the other thing you said?
Yeah, that was basically it. Or just with molasses and that.
Well, molasses is bacterial, so just be careful with that. And there was something what was the last bit you asked me?
Oh, that was about it. Yeah, that's about it. Just should our systems be more fungi dominated? So.
There was something else you said though. Oh, you did. Thank you very much. I knew there was another important part. Can you stimulate the wrong microbes? Yes, you can. So obviously with molasses, too much molasses, you can stimulate. I mean, if you put molasses on, you can get plants will grow more, but they'll grow more because the molasses is stimulating bacteria in the soil that break down soil carbon and release all the nutrients that were in the carbon. So you get all the trace elements and everything that were in those humic polymers. And yes, it can be rocket fuel, basically, but you're going to destroy soil structure, and then over time, your soil's going to deteriorate. So you might get a short-term benefit from it, but it's not going to be good long-term, especially if you're looking at soil carbon levels. So that's just something to be aware of. Yes, you can definitely stimulate the wrong microbes. And things like using raw manures or something, if they haven't been composted, that kind of thing, you know, that can be detrimental as well.
Yep.
For sure. You definitely can stimulate the wrong microbes, but if you use something that's got autoinducers in it, in other words, the chemical signature of microbes from a good environment, like a vermi-liquid environment or a fermented seaweed environment or something or other like that, what you're doing is stimulating the plant to feed the microbes, and what comes out of the plant will be the right things, because the plant will send signalling molecules as well as food to the microbes that it wants. So really what we want to do is to stimulate the plant to feed the microbes that it wants, because then we're using plant intelligence, not ours. They're smarter than us in terms of knowing what microbes they need around their roots and what minerals and trace elements they need. The plant knows, and it will signal to the microbes to get those. So that's what we actually want to do, is stimulate that process.
If you're looking to do stuff with your soil, like improve it and to find out what you're lacking in, whatever, not to obviously add synthetic fertilisers or something, but certain plants can add additional stuff. Because through some of the reading, all soil tests aren't the same sort of things. So what's the best soil test? And also, what should we be looking for in a soil test?
All right, I would use the soil test to look at things like cation exchange capacity and carbon level, total nitrogen level, total phosphorus level. And I wouldn't really probably use it for anything else, really. If I wanted to know whether is this for a crop or for a pasture, I'd actually be looking at leaf tests.
Yep.
To see what, because I've seen so many times, I've seen thousands of soil test reports, and I've seen, like in New Zealand, we see a classic case of an inverse relationship between Olsen P, which is the test that they use for available soil phosphorus, and leaf phosphorus levels. In other words, if there's really high Olsen P levels in the soil, you'll get low levels in the leaf. And you think, how could that possibly be? It's because really high levels in the soil are going to switch off mycorrhiza, which means that the whole uptake mechanism has been interfered with. So there's lots in the soil, but it's not getting into the leaf. So what use is the soil test, if you see what I mean? Other times there can be really low levels of P in the soil, but you measure the leaves and it's perfect in the leaves. So you think a low level in the soil would make you want to add it. But then if you add it, you muck things up. So I'd be really careful, like with interpreting a soil. You almost can't interpret them really. Total P, just to give you an idea of really, and the thing is, total phosphorus is not total phosphorus anyway. Total phosphorus is what is available with a concentrated nitric acid extraction. And I've seen total phosphorus levels over time go up, where we've gone every year and taken a soil test. Colin Size is a classic example. Colin Size, some of you may have heard of the guy, the pasture cropping guy. We're measuring his soils every year and total phosphorus was going up. The first year I thought, oh, well, you take soil from a slightly different spot. I mean, it's a different soil sample, right? Can't ever be the same one. So maybe it's just because of that, sampling variability. And then the next year it had gone up again, and the next year it had gone up. So I rang the lab and I said, so either your extraction method's changing or something funny going on here. I said, I would have thought total phosphorus was total phosphorus. And he goes, oh no, total phosphorus is what we can extract with nitric acid. I said, well, can microbes in the soil get more phosphorus than what you can get with nitric acid? And he said, yes, of course, like as if I was really dumb. And I went, oh, okay. So total phosphorus is probably only like 75% of the phosphorus that you've actually got. But if you do total phosphorus test and work out how much you've got and how much goes out the gate every year, depending on what kind of production system you've got, you're going to be selling off phosphorus in your produce, right? It's going to be a fraction of what you have in total. And you'll find that nearly everybody who's calculated that you've got enough phosphorus for at least 10,000 years, even if it was going out the gate at a massive rate every year. If you were doing a research project and you wanted to know whether carbon was increasing over time or cation exchange capacity was changing or something, it will be worthwhile. Or if you were really concerned about phosphorus and you wanted to know for sure so that you could sleep at night that it's okay not to put phosphorus on, then you could do a soil test and find out what your total phosphorus level is. Otherwise, I don't think they're worthwhile, because they're only going to tell you what's in solution, and what's in solution is not how plants operate as a general rule. If they're in a microbially rich environment, they're going to be communicating with microbes all of the time to get the things that they need.
Christine, you might have already answered this question, but is the accumulated amount of phosphorus that we've put on through super and triple super over the years going to be an impairment to repairing the soil, getting it back to a biological state?
Oh, that's a good question, David. Thank you for that question. The answer is no. It's not going to be an impairment because, unfortunately for you at the time, it was all pretty much fixed after you applied it. So within a couple of hours or a couple of months, depending on how moist and warm the soil was, the phosphorus that you put on went into some kind of a compound with iron or aluminium or manganese or something and got taken out of solution, right, out of the system. So it's there. So now, if you have applied those things in the past, you have a huge phosphorus bank that your plants can tap into by, again, if they send the signalling molecules to mycorrhizal fungi or phosphorus solubilizing bacteria to actually get that. So you haven't lost it. Unless you lost topsoil through erosion, So if you had like water that's got sediment in it moving off your farm, if the water that's running off your farm in a high rainfall situation has got mud in it or sediment in it, then you are losing phosphorus. But if you've got clear water running off the farm, you're not losing phosphorus. So all the phosphorus that you've applied is there, and you can access it with microbial activity. And it's not a detriment at all. Luckily, it does get tied up in the soil pretty quickly.
Okay.
But the problem with it is that people will put it right under the seed of a newly germinating plant, and that stage it hasn't been locked up yet, and it is water soluble and it will interfere. That's where it's a danger when it gets put under the seed.
Thank you.
Christine, could I just add a couple of things? With soil samples, I tend to agree with you up to a point. The thing I like about soil samples, and particularly for a lot of you farmers that have been taking them for a lot of years, they're an excellent way of actually seeing the big picture. So one soil sample, you might as well treat it like a grain of salt, because you tested so little bit of that soil across that paddock. But if you've got a soil sample out of that paddock year on year on year, over a decade, for example, you can then see what the trends are. So they're very good for an overall big picture of the soil that you have. So don't throw your soil samples away, and don't necessarily stop soil sampling, because it's a good way of looking for trends, particularly if you're trying to improve. So increasing organic carbons or CECs, those sorts of things. Another thing to remember is, with things like phosphorus, the main reason why I started with sunflowers, for example, they're what's called highly mycorrhizal. So different plants, and Christine, you've said that, different plants are associated with different microbes. So sunflowers, being highly mycorrhizal, actually encourage mycorrhizal fungi, which is one of the key biological mechanisms that unlocks phosphorus, for example. So that's why I started with sunflowers, was I was trying to create more mycorrhizal fungi and unlock some of my locked up phosphorus. Now, I have not established whether or not I'm actually unlocking that phosphorus, but I have actually, through DNA analysis, determined that just by planting sunflowers, I can increase not only the number of mycorrhizal fungi that are in my soil, but the number of different varieties, if you will, substantially. So different plants encourage different microbes, and that's one. Like we know, legumes are associated with rhizobia, sunflowers with mycorrhizal fungi. Sorghum is another one that they now know actually, denitrification inhibitors. So sorghum can actually exudate a compound that slows down the loss of nitrogen, particularly in wet ground. So it knows that it will need that nitrogen later. It doesn't want it now. So it actually releases a compound that allows the bacteria to slow down that denitrification process. So it biologically holds nitrogen in the soil. So science is learning more and more all the time about those relationships between plants and different microbes that do different jobs.
So the comment that you made, Simon, about the mycorrhiza and the phosphorus, that phosphorus can be actively transported by mycorrhiza who will take carbon from the plant to colonies of phosphorus solubilizing bacteria that are at their hyphal tips. In other words, at the end, like you've got that hyphae running out to the soil, and then at the end of that, there'll be a colony of bacteria that are able to solubilize phosphorus. They'll be getting fed with carbon coming through that fungal hyphae. And so that, if you were to do a soil test, you're still not going to pick up that phosphorus because it's in an unavailable form until the bacteria get that carbon, and they will only solubilise what the plant needs. So you're only going to pick it up with a leaf test.
Yeah.
In other words, even having more mycorrhiza in the soil is not going to increase what shows up on a soil test in terms of available P necessarily. Although it could increase your total phosphorus, and that's probably what happened with Colin Syers with his total phosphorus going up every year. It was probably because he had more mycorrhizal fungi in the soil that and they've got phosphorus in their bodies as well. So even when you have like more microbes in the soil and you do a soil test, you're going to end up with some of the microbes in your soil test. And if they're full of nutrients, they're going to show up on your soil test too. But it is very interesting, the whole thing. The problem is that we've tended to think of all systems really, like even the human body. What happens if you're sick and you go to see the doctor? They'll take a blood test, or they'll listen to your heart, or they'll do all these things. So a blood test is going to be like chemistry, right? Or they'll do a physical thing like listen to your heart or feel your pulse or do something like that. But like I said before, are they actually looking at your gut microbiome? Are they looking at the microbes that are running you? And we need to think about the measurements we're taking of soil. Are we actually looking at what's running that soil, or are we just doing some chemical and physical tests? Because they're not really going to tell us a lot, unfortunately, about what's happening with the soil microbiome.
Okay, can you, the last one, make it quick?
We've talked about phosphorus and nitrogen. What about other minerals like zinc and boron? When they're deficient in the plant, what can we do to add that then?
Okay, so one of the main things that mycorrhizal fungi bring back to plants is zinc. So the most important element that mycorrhizal fungi bring to plants is phosphorus. Well, probably the most important thing that mycorrhiza bring to plants is water, and then after that phosphorus, and then the third thing is zinc. So if you don't have those mycorrhizal relationships, your plant will be zinc deficient, and we see that like across the wheat belt particularly. I don't know much about this part of the world, but in the wheat belt, we see where people have used MAP or DAP to grow wheat, their plants will be zinc deficient. The soil wasn't deficient in zinc, but they've interrupted those mycorrhizal linkages and the plant can't get the zinc that's there. So if they stop using MAP or DAP, start using a biostimulant, your plants, I've seen zinc levels go up 15 times, like a factor of times 15. Incredible in plants when people have just stimulated the biology in the soil.
Yeah, well, we haven't used chemical fertilisers on our place for over 4 years now. All our leaf tests, even when we beforehand and even now, are well up in the phosphorus and nitrogen. We wasted money before that. We've proven it to ourselves a dozen times over. With phosphorus and nitrogen, in the leaf tests, they're always up. In our soil, we're a little bit down in zinc. We're always a little bit down in our leaf tests. Boron, when you're producing fruit, is a very important thing, the uptake of calcium, and for the stop fruit being deformed. How can we increase it? Through
okay, so if you actually have deficiency symptoms of boron, if you have deficiency symptoms in your plants,
there is
I would definitely recommend adding some because it's not a big deal.
Well, we foliar add them, and that brings it up in the leaves.
Yeah, so like 1 kilo per hectare or something like that of soluble or something.
Yep.
Look, if you've got actual deficiency symptoms showing up, and you know that you have to apply it at a very small rate because it's toxic in high levels, there is nothing wrong with applying any of those trace elements if you need them. The only thing that switches off the communication between plants and microbes is nitrogen and phosphorus. They're the only two elements that do that. The other things don't interfere with communication at all, but you do have to be very careful with things like trace elements like boron and zinc and copper, and there's a very narrow range. Below that, they're going to be deficient. In that range, it's going to be perfect. And if you go over that, they're going to be toxic. So boron will be toxic if you put too much on, but provided you know what the right amount is, and I think you don't apply it every year, is that right? It's every couple of years or something, they'll say, with boron.
Boron's one of the most leachable things going.
But anyway, if you need it, apply it, and it's not.
We apply it fully.
You're going to be putting on 1 kilo per hectare or something like that. Yeah, very, very small amounts.
Negligible.
So yes, if you need them, if you need to trace elements, apply them. Definitely. Like the plant can't function if it doesn't have sufficient boron.
Yep.
But what kinds of plants are they?
Mangoes.
So what do you have growing underneath them?
Pardon?
What do you have growing underneath them and around them?
We've got grass, mainly.
Okay.
Grass and legumes, and very diverse in it.
Oh, it is diverse.
Yeah. We've also had all our microbes tested by MicroWise down in University of Adelaide there. Mm-hmm. They've all come back fantastic. And microzonal fungi, the carbon fixing things, we've increased from surrounding area was 0.6 and 0.8, which is pretty common for Mackay. For organic matter, we're up as 4.2s.
Fantastic.
3.6s.
Okay, so you've got living ground cover under your plants. You've got a diversity of living ground cover.
Worms have come back that, you can't walk across the place without walking on vermicast. But we're still having troubles trying to get, as I said, certain minerals back up. And we're folio attacking them at the moment, but I was just
I think what you're doing is...
anything you could suggest that we could plant that may increase microactivity to increase that in the plant,
Well, I mean, it
soil.
could be possible that for some reason you could be in soils that don't have much boron. And boron is a really highly mobile element.
Yep.
And I don't know, are you irrigating your plants?
Yes.
It will, that's just going to wash boron. It's going to take boron out of the soil profile continuously. So in an irrigation system, it might be that you do have to apply boron. And for trees, they're obviously going to need a lot. So there's nothing wrong with applying that if you need it in small amounts. Absolutely nothing wrong with it. In fact, you really need to, because if you don't, as you said, you're going to get deformed fruit and a whole lot of other things will not be right if you don't have enough boron. So yes, leaf tests, and use your leaf tests as a guide as to how much the plants need, and yes, foliar applied boron if you need it, definitely.
Yeah, well, we put biostimulants through our fertigation system as well. Such as worm juice or fermented ones.
So sometimes, under irrigation systems, because boron is highly mobile, you will lose it. So there's nothing wrong with applying it because it's being leached out of the system.
That's why we apply foliar, you know what I mean? Thank you.
Sorry, I have to cut you short, Ken, I'm sorry. But I cut already into your afternoon tea session, so there's a couple more days with Christine. I think some of you are joining us. Yep, I got some nods, and I think we might have to re-invite Christine and join us some other time because, a lot of questioning